Thank you for the introduction, and thanks a lot for the organizers for this amazing event. So I'm a postdoc at UPenn, and my advisors are Andrea Liu and John Crocker, and this is our work together with our collaborator, Marco. So I realized that the formal title includes a mouthful of terminologies like rigidity, homeostasis, and mechanosensitivity. So I wanted to start from more like accessible title. How does a cell stay mechanically rigid even when its architecture constantly changes? So a bit more about myself. I am part of this vibrant community at the Center of Soft and Living Matter at Penn. So we study anything that's soft matter or biology. And last year I graduated from Princeton, where I studied statistical mechanics and chemical physics from Salto-Cato lab, and now I'm postdoc at Penn. And at Penn, I'm gearing more towards biophysics and system biology from a perspective of computational modeling and theoretical physics. So I'm interested in generally algorithms that cells live by, or how cells behave if we consider them as engineers. So the talk that I'm going to present is considering the cell as a mechanical engineer. So before we go into cells, I want to start from a more broad perspective in terms of metamaterials. So metamaterials are natural or engineered materials whose property not only stem from the atomic or molecular composition, but also stems from the connectivity or the spatial arrangement on at least the nanoscale or nanometer or micrometer level, or much larger. Some examples include 3D printing materials, knitted fabrics. It doesn't matter which yarn we are using to knit the material. As long as we are doing the same kind of stitches, we have similar elastic properties. Also, origami, kirigami, doesn't matter which page we are using. It matters how we fold them. In terms of biology and chemistry, we have examples like hydrogels, where we have a lot of filaments that are stapled together by crosslinkers. And the biological variant of the hydrogel is the star of today, actin cortex. So that is a network of biopolymers that lie just beneath the cell membrane. If the cell just contains water and lipid, it will be too soft. So this actin cortex that underlines the cell membrane, it supports the cell shape, makes it more rigid, and it also regulates all kinds of cell mechanical processes, including migration and mitosis. So the actin cortex, compared to a passive hydrogel, it is highly dynamic, and it maintains rigidity in a crazy way. So the first cool things that it does is that if you look at the average number of filaments joined at a crosslinker, it's either 3 or 4. So the average coordination number is between 3 and 4. And that is below the Maxwellian isostaticity that says you need 6 filaments per crosslinker to stay rigid, because you need to constrain up and down, left and right, front and back in 3 dimensions. But the cell does it with fewer coordination number. And that is because the filaments are undergoing some pre-stress. So as a filament is incorporated into the cortex, it is already pre-stretched. And this pre-stress shifts the floppy to rigid phase transition to smaller coordination number. And from this, you may think, wow, so every filament is mechanically extremely important. So we shouldn't touch any of them because it will go floppy if we touch them. But it turns out that the filaments and crosslinkers are undergoing fast remodeling or turnover. So for every 30 seconds, the actin cortex in your cell has undergone a complete turnover. So that is highly counterintuitive. How does the cell really maintain rigid, but is also highly dynamic and sustains such fast turnover? And also this turnover can take up to 50% of total ATP hydrolysis. So why does the cell do that? And how is it possible? So it turns out that we have some clues of how this happens. So our collaborator, Marco, in his physical review research last year, we have found that there are a lot of mechanosensitive dynamics. So the addition and pruning of filaments and binding and unbinding of crosslinkers are far from completely random. So if we just do random pruning of filaments, the rigidity of the actin cortex will be destroyed even at coordination number greater than 4. So we know that the pruning is not completely random. But if we do something called tension-inhibited pruning, that is, we prune the filaments under small tension first, then the cell decides, it is like the cell decides that, oh, this filament is not useful for my rigidity, so I may as well cut it and recycle the material and put it somewhere else. And if we do this tension-inhibited pruning, then Marco and Andrea and John have found that the filaments preserves rigidity for coordination number below 4. Yeah, but we don't really know how the filaments should be added onto the network. And also, as a parallel to filament dynamics, we have crosslinker dynamics. And here is a completely parallel idea that we have catch bonds. So for a regular crosslinker, if you put too hard on it, it breaks. But for such catch bond, if the force on the crosslinker is too small, then the crosslinker splits apart too. So it has been suggested that such catch bonds are useful to build stronger networks. And indeed, we have found enzymes and proteins that exhibit both tension-inhibited pruning and crosslinker properties. However, the mechanosensitivity, how does the mechanosensitivity incorporate and contribute to rigidity homeostasis is not fully known. So the goal of this work is first to understand how tensional rigidity of the actin cortex can persist amongst complete structural turnover. And more precisely, we want to understand the role of mechanosensitivity or mechanosensitive local rules in rigidity homeostasis. And we want to use a computational approach to study how does the rigidity homeostasis arise from a minimal set of local mechanosensitive local rules. And lastly, once we have built such models, if we can build them, then we want to study the topological and geometrical properties of such models because we are mathematicians. All right, so here's some description of this model. We are modeling the cortex as a spring network. So here, each actin filament is modeled as an edge that is a Hookean or harmonic spring, and each crosslinker is modeled as a node that binds the filaments together. And the incorporation and pruning of filament is modeled by addition and deletion of edges. And binding and unbinding of crosslinker is modeled by splits and merges of nodes. So a node of degree z can be split into two nodes of degrees z minus 1 and 1, and vice versa. And here we are assuming that we are in the tension-dominated regime, so no bending rigidity, although that can be important when we fine-tune this model. Okay, so we have tried a lot of dynamic ingredients, and here is what works. It needs basically 4 ingredients. So we have built 2 models, one for edge dynamics and one for node dynamics, just to separate. So for the edge dynamics, we first need tension-inhibited pruning, as we have just said. So that means an edge under low tension below some threshold, theta E for edge, is always removed in each iteration. And secondly, in each iteration, we also have a small fraction of random pruning. So an edge can be randomly removed with a small probability, regardless of the tension on it. And of course, the opposite of pruning is edge insertion. So we are inserting pre-strained edges by which an elastic energy is injected into the network. So to do that, we choose a nucleus in space, shown by the red dot, and we prescribe a spherical region. And if 2 nodes of degrees 3 or less are fall inside the nucleation region, then we add a pre-strained edge between those nodes. And lastly, because we are using phantom edges that can pass through one another, we want to compensate for that. So we are adding a steric repulsion that says, and for a new nucleus, it is more likely to be added to a less crowded region of the space. So we are adding that with a Monte Carlo approach. Yeah, and parallel to that, we have a dynamic node model. So that means for each node, it can be split if the force on one of its, if the pulling force on one of its edges is below a certain threshold. And we also have a small fraction of random disassembly, where a node can split regardless of the tension on it. And of course, the opposite is edge merging, node merging. All right. So a degree 1 node can merge with its nearest degree 3 or less node in a capture sphere. Yeah. And lastly, we have a steric repulsion, because degree 1 node can swing around, and we want the edges to form large angles rather than small angles. Okay, so this is our model. And I wanted to add a little bit technical interlude about our infrastructure for elastic networks, because these are highly ubiquitous things in biology and civilizations alike. You can model the actin cortex as an elastic network, but also a knitted fabric. So because of that, I developed a package that can treat them more generally. So for our network, elastic networks data structure, we have a graph that tells us the connectivity between the nodes in the network, and of course, the basis that tells us what is the simulation box, and the points, the locations of the node in space. And for our each edge, we have rest length and Young's modulus. And because we are in periodic boundary conditions, we also specify which image of node J node I is connected to. And with this package, we can create and modify network. For example, I have created a lattice network, and we can look at what are the edges inside the network, and we can prune certain edges. And we can also compute the elastic moduli of the network with automatic differentiation. So that gives you the exact elastic moduli. Cool. So now we have the results for rigidity homeostasis of the actin cortex. So first, I will show results for the edge model. So here is a cool video that shows the edges are appearing and disappearing. And we have exponential decay of edge labels. So whenever an edge is pruned, its label changes. So the fraction of unchanged edges is decaying exponentially, and we have a typical turnover time. So this is shown by the red curve. And for the blue curve, that is a hypothetical turnover if there's only random pruning without a tension-inhibited pruning. So we see that both random and tension-inhibited pruning is contributing to the edge turnover. And this model indeed reaches a dynamic steady state. So rigidity homeostasis means we have constant steady state of rigidity in terms of the bulk and shear moduli and elastic energy, but at the same time, we have a complete structural turnover. So we have achieved that. The mechanical properties reach steady state, and we have exponential decaying fraction of unchanged edges. The story is similar for the node dynamic model. So here's the video. You can see that the edges are persistent. The nodes are merging and splitting, and the edges are taking turns to carry load. The darker the color, the larger the tension on the edge. Yeah, so we have exponential decay of unchanged node labels. And again, the upper bound, it is bounded up by the hypothetical decay if there's only random splitting of nodes. And we have steady states of mechanical properties. Cool. And and not only mechanical properties reach steady states, we see that the structural properties also reach steady states. So here we have the structural factor, that's the Fourier transform of the pair correlation function between the edge centers. And we can see that for T1, the time interval, and T2, another time interval that is separated by 5.2 times of the turnover, typical turnover time scale, we have statistically invariant structural factor. And also for the same two time intervals, the cumulative density distribution of edge lengths is also invariant. So this is almost like an equilibrium, but energy is constantly being injected and flowing out. So it's a dynamic steady state. Cool. And you may wonder whether we need all four of those ingredients in our models. And we show that, yes, those are indeed minimal. So for example, if we take away the random pruning, and if we just have the tension-inhibited pruning, then the network fails to turn over. So when there are about 93% of still 93% of unchanged edges, the network stops evolving. And on the other hand, if we take away the tension-inhibited pruning, and if we just have random pruning, then the network loses rigidity very quickly. The story is the same for the node model. If we don't have random pruning and random node splitting, then the fraction of unchanged node it stalls at about 60%. And the network will lose rigidity if we don't have force-sensitive splitting. So now we have really identified what is needed for rigidity homeostasis. And what's really surprising about this model is that the network constituents, including nodes and edges, they undergo diffusion as in a liquid. So this means there's no permanently protected local structures or correlations, because everything is really destroyed and rebuilt. And the model also captures the viscoelasticity. It means if you poke the network, then you feel a resistance. It means the network is elastic, but also the edges and nodes are undergoing diffusion, as shown by the linearly increasing mean square displacement. So the network constituents are also diffusing as if in a liquid. So we have both elasticity and viscosity. And this shifting microscopic encoding for steady global function is known as representational drift that has already been found in, for example, the visual cortex of the mice neuronal network. And here we are finding that this individual cell is doing representational drift too. Cool. And what's really cool about our brain, the neuronal network of our brain, is that it can heal from severe damages. So if a certain part of the brain has been damaged, sometimes the brain can still function properly. So we are also finding that for the elastic networks in the actin cortex. So to give it a severe damage, I created a tensionless hole where everything is taken apart within a hole that whose diameter is a third of the box side length. So that's a really large hole. And after 8,000 iterations, it completely restores to the previous steady state. So here is a video where we see that there was initially a hole that has been recovered, healed. What's cool about that is that the mechanical property recovers much faster than structural property. So if we look at the elastic moduli, the bulk and shear moduli, and elastic energy, they restore to steady state within 1,000 iterations. Now, however, the structural factor and the number of nodes in the network completely restores only after 4,000 iterations. So it's as if the cell is deciding that, oh, its mechanical property is more important, so we fix that first. And then only after that, we fix the topology and geometry. Yeah. So next, because we are mathematicians, we want to study how does the actin cortex behave as a graph theorist. So that is very interesting. So to do that, we want to compare our actin cortex model with a hypothetical model that has similar number of nodes and similar number of coordination number. So we compare the diamond network with our actin model. And we find that even if they have similar values of z, one is 0.92, one is 4, the actin model has a higher shear modulus. But you look at the trade-off between bulk and shear moduli. It seems that the actin model worries about shear modulus more. And this also happens with the dynamic node model. So because these models have similar coordination number and similar volume fraction, we believe that the key difference comes from the topology. So we use graph theoretical metrics to determine what happens. What is the difference between their topology? So the first thing I thought about is that because triangles resist shear, so we must count the number of triangular loops. And indeed, we find that in our actin cortex model, the number of triangles are larger than for the diamond, which contains none of the triangular loops. So that's true. And also, we look at two centrality metrics. The first is closeness centrality. So that is the reciprocal of farness, so to speak, or the reciprocal of the sum of the shortest path lengths between a node and all other nodes in the graph. We find that the mean closeness centrality for the actin cortex models are larger than those for the diamond. And we also look at the betweenness centrality. So that is the degree to which nodes stand as a bridge between the shortest path from another node to a third node. Surprisingly, the mean betweenness centrality for our actin cortex models are smaller than those for the diamond. So that's, in summary, there are many more triangles in the cortex model. And topologically, the cortex model are denser, even if their volume fractions is the same as the diamond. And the passes between nodes pass through fewer bottleneck nodes in the actin cortex model. So this may contribute to the fact that the actin cortex is really resistant or robust to damage because there are fewer bottlenecks. So I really, I would like to see many experiments done to analyze the actin cortex. So in summary, we have found elements needed for rigidity homeostasis include mechanosensitive disassembly, a small fraction of random disassembly, and energy injection upon assembly. And we are able to use graph theoretical metrics that allows us to elucidate how cells exploit topology to build stronger and more resistant biopolymer networks. In the future, we will study the actin cortex as a learning metamaterial and see if it can remain rigid under fluctuating stresses. I would like to thank my advisors, Andrea and John, and our collaborator, Marco. Thank you very much.
Thank you for this very interesting talk. So maybe my question just continues from your last comment. I was wondering if any of the measurements that you have made can be compared to have been compared to experimental data.
So we have compared. So we have compared with experimental data mainly in a qualitative way. So we are not yet considering quantization. So one of the things that we have compared is called strain stiffening. So we are stretching our model to some finite strain. And in experiments, people have found that the elastic moduli increases when we give it an external strain. So we have seen that. And we have also looked at the diffusion of our nodes and edges and how it correlates in space. So this is given by the Van Hoff's function. And in experiments, people have found that the Van Hoff function is fat-tailed, actually. It's given by a Cauchy distribution, but truncated by exponential decay. So we have also found that in our models, this exponentially truncated Cauchy function is able to describe the diffusion. Thank you.
Excellent talk. Question, how much damage can your network recover from? So you talked about a big damage where a big chunk was taken out, but I can imagine that you have different places where you have damage. So have you investigated how much damage both disconnected or in one place, if you make it larger or smaller, your network can recover from?
So that's an excellent question. So this is one of the two experiments that we have done. So we find that if the hole is a little bit larger, like 0.35 of the box side length, then it loses rigidity. And another experiment we have done is to increase the probability of random splitting and randomly destroying the filaments and crosslinkers. And this is actually our models are very robust to that. If we increase the fraction of random splitting by 50%, it still remains rigid. But if we increase it to twice of this, what I have shown, it loses rigidity. Mhm.
Hi.
Yeah. Hi.
Sorry. Yeah. So it's really amazing talk. I really loved it, especially the idea of representational drift of how neurons represent things and how the actin cortex seems to be working. But in neurons, we do have some idea of what the neurons are representing, like some features of the stimulus or something that the brain is reacting to. Do you have any hypothesis about, like, so we do have a fixed goal function of maintaining rigidity, but what might be the network trying to represent in some way?
Right. We think the input and output relationship is the relationship between strain and stress. So given external like a squeeze or shear, what is the increased amount or decreased amount of elastic moduli or the stress itself? So yeah, there are definitely many other input and output relations we know of, such as the pooling of the motors that we will study in our next paper. Yes.
Thank you very much. I think we should move on.
Thank you very much.